Good morning, everybody. My dear student, colleagues, and all the viewers who are watching this program live from Facebook page and YouTube channel. So I'd like to welcome you all to our international physics webinar. So this is our 276 international physics webinar. And those who are watching from Canada and USA, I'd like to say good morning. And for this part, good evening. So I think you are well, and we have organized this webinar after about 1 month. So thanks for supporting this throughout these 2.5 years. So today we have with us Dr. Leonid S. Brown, Professor, Department of Physics, Associate Dean of Research and Graduate Studies, CEPS, University of Guelph, Ontario, Canada, and he has already connected with us. So I'd like to welcome our speaker, sir. Good morning to your part and good evening to here. So thanks for joining with us.
Morning.
And he will talk on the manipulating and studying proteins by light. I think you will enjoy. Sir, it's your time. You can start your session. And the student and viewers, you can join with us directly using the link. I'll send the link in the comment section. You can also ask question by commenting. So please enjoy this session.
Thank you very much. Thank you for the invitation. And everyone, thanks for coming. Good morning, good afternoon, and good evening. Today, I'm going to talk a little bit about our research. I will give you an overview of exciting light-activated proteins, and we will discuss how they can be studied with physical methods. So I will introduce you to our methods, and I will give you some examples. And maybe my talk will combine biology and physics and some chemistry as well. So hopefully it will not be too much for people who are pure theoretical physicists. I hope it can be still interesting because it connects to life, it connects to our bodies. So I'm welcoming you from the University of Guelph. Guelph is a beautiful town in Ontario, not far from Toronto. So let's talk about proteins. So you probably know that most proteins do not absorb visible light. They are colorless, but there are exceptions. And the most striking exception you're all familiar with are proteins responsible for our vision. We all have them in our eyes, and they're called visual rhodopsins. So in our eyes, in our retina, we have special photoreceptor cells, rods and cones, and they contain rhodopsins, which are activated by visible light and allow us to see things. What is the trick? Why most proteins cannot absorb light, but rhodopsins can? This is because they have attached small molecules, so-called chromophores, which give them color. And for our visual rhodopsins, chromophore is a retinal, this small molecule shown here. When we see light, the retinal changes its shape, and it is translated to electric signal, which our brain perceives as light. So from point of view of physics, our visual systems are amazing because they are single photon detectors. We can see single photons. Why? This is biochemistry. We are not going into the details, but the trick is that activated protein molecule, activated by single photon, can amplify its signals through complicated biochemical cascades, which are shown here, through a number of chemicals inside our cell, which ultimately translate into electric signal because they change electric potential on our membranes. And this is translated to our brains. So this is probably the most important light-sensitive protein for us. What my group studies in Guelph and many other people study are very similar proteins. They are also called rhodopsins, but they are not human. They don't come from animals. Instead, they come from microbes. They come from bacteria, fungi, and algae. So this is a good time to introduce rhodopsins. In general, basically, they all have similar structure. They all have 7 helices. They're sitting in our membrane, so they're membrane proteins. They're sitting in cellular membrane, and they have 7 helices, which surround the molecule I mentioned, the chromophore which senses light. And so I introduce you to the type 2 animal rhodopsins, which have visual functions, but I will be mostly talking about their counterparts in microbes, which have the same structure, similar photochemistry, but very different functions. They are used for bioenergetics because they serve as ion pumps, and they also use for photosensory functions because they can sense light and translate it into different intracellular signals. So they can be channels, they can be enzymes, they can be other sensors. So a little bit of history. So here is a typical microbial rhodopsin, and this is the first microbial rhodopsin discovered back in 1972. So we have 50 years of research on microbial rhodopsins, and this microbial rhodopsin, a prototypical one, is called Bacteriorhodopsin. So you can see it has 7 helices, it has retinal chromophore, and when you work with it in the lab, it looks very beautiful, like this. This is a picture from my lab, and physicists love working with it because it is colored, so you can see it. And second, it's very easy to get in large quantities. It is very stable, so there is a lot of physicists working on this protein. So a bit of a history. It comes from so-called halobacteria. So halobacteria live very interesting bugs. They live in 5 molar salt. So in salt lakes, salterns, you name it. They are extremophiles or holophiles adapted to very high salt conditions. So when when there is enough light, but not enough oxygen, it was noticed that this bacteria, which can swim, by the way, form purple patches. These purple patches were called purple membrane, and these purple patches turned out to be 2-dimensional crystals, native 2-dimensional crystals of Bacteriorhodopsin. And Bacteriorhodopsin is a proton pump. It pumps protons outside of the cell when it absorbs light. I should mention that this is very important bioenergetically because gradients, transmembrane gradients of protons, serve as energy sources for us. We can convert them into ATP, as shown here. And ATP, as you all know, is biochemical currency of the cell. So back in the 70s, there was a lot of research, and later on as well, and which very quickly discovered the structure, interesting structure of this Bacteriorhodopsin. It forms these 2-dimensional crystals, lattices of trimers, and I'm showing you some details of this structure. By the way, this pioneering work on 2D crystals several years ago garnered a Nobel Prize for electron microscopy work. So let's talk a little bit more about the details. So this is the chromophore. This is the chromophore, the retinal I mentioned. It is attached to the protein through the so-called Schiff-base linkage, which is carbon to nitrogen double bond, where nitrogen is protonated. So it is included in some first-year physics books as a typical molecule because it has alternating double and single carbon bonds, which create a pi electron cloud, which absorbs visible light. Because of these 6 conjugated double bonds, it can absorb light. Very interestingly, upon light absorption, the shape of retinal changes. It rotates around the C13-C14 bond very quickly, and this is the act of energy storage. And we will discuss a little more, but first I want to briefly mention that the color to which rhodopsins are sensitive can be tuned. Just like in our eyes, we have different visual rhodopsins responsible for perception of different color, for our color vision. Microbes can also tune their absorption depending on what kind of light they want to sense. Depending on where they live, light will be different. So they want to tune the spectrum. So here we are going into spectroscopy right away. There will be more spectroscopy. So here I'm showing you how absorption spectrum, this is visible absorption spectrum of retinal, changes in vacuum, in solvents, and then in proteins. So proteins can tune it by strategically placing charges around it, also by twisting the retinal, and also by introducing water molecules around it. Great. So let's go back to the isomerization. I told you that upon absorption of light, so-called photoexcitation, the isomerization is extremely fast. But first of all, some of you may wonder, on the previous slides, I showed that the rotation is around the double carbon-carbon bond. And those who studied chemistry will tell me it's impossible to rotate around the double bond. The barrier is too high. Indeed, here is the diagram of the ground state of retinal in bacterial rhodopsin and the excited state. And indeed, in the ground state, the barrier for rotation is extremely high, so we cannot isomerize. But in the excited state, after we absorb light, the situation is completely different. The energy surface is different, and we can rotate and isomerize the retinal. And it happens very fast. You can see 500 femtosecond is extremely fast. And first, when it was discovered, some theorists were skeptical that you can actually isomerize the molecule so fast, that you can move heavy atoms so fast. But it is true. It does happen, and sometimes even faster than 500 femtoseconds. Okay, so the next step, the retinal isomerizes. Here I'm showing you X-ray data where this isomerized state is trapped. And when it isomerizes, it stores some energy, and it no longer fits into its binding pocket. So it starts pushing on the protein, and the energy is transmitted to the rest of the protein from the retinal. And then proteins perform their various functions triggered by retinal isomerization. This is so-called photochemistry going into photobiology, photochemistry and photophysics. So what happens as a result? The stored energy is slowly dissipated. And when it dissipates, it goes through a number of thermally metastable states, which we call photointermediates. And these states are distinct spectrally, so we can distinguish them by their color. Different spectra, you can see some more yellow, some more blue, some more red. And they also distinguish kinetically. Some form on picosecond scale, some form on microsecond, some form on millisecond. So apparently this is a very good ground for doing all kinds of spectroscopy, including time-resolved spectroscopy, which we do. So kinetic spectroscopy to follow these states. And luckily, these states correspond to important biological events, such as proton transfers, conformational changes, changes in the retinal geometry. And we can all follow this by spectroscopy, as I will describe very soon. But first, let's talk a little more about the functions. So the energy is passed to the rest of the protein. It performs various functions. What kind of functions we are talking about? Here is some overview. So the classical function I mentioned for bacterial rhodopsin is pumping protons outside. Back in the 70s, there were similar proteins discovered which pump chloride ions inside. In the last decade, there were similar proteins discovered which pump sodium ions outside. Then there are all kinds of photosensors, which are activated, interact with their transducers, and then regulate swimming of bacteria, or they can regulate DNA transcription, or they can be channels, light-gated channels. The light opens the channel, ions, cations or anions, depending on the kind of the channel, rush in or out, change electric potential, signal as well. Finally, fairly recent discovery in this century that rhodopsins can be linked to enzymes, so they can serve as light activatable enzymes which can produce various signaling molecules inside the cell. So there is a huge variety, and we are not done. There is a lot of work looking at new species of rhodopsins, discovering new functions. So it's a very exciting field. It turns out that in this century, there was another major development, why this field became extremely exciting and was sort of revitalized. Because a lot of neuroscientists got really interested in these molecules. Why? Turns out that you can introduce these rhodopsins into neurons, into nerve cells, and you can regulate neural activity with light. This is so-called optogenetics, and optogenetics is extremely popular now. All neuroscientists now love biophysicists, they love rhodopsins, so we are in business. All these rhodopsins have high potential to serve neuroscience and biomedicine, right? Imagine the potential where you can use these molecules to activate or to suppress neurons with light, right? So you can study our nerve systems, you can regulate it, you can maybe treat some diseases, because some diseases are uncontrollable firing of neurons. And using these rhodopsins, you can suppress this uncontrollable firing, such as in epilepsy or Parkinson. Or, on the other hand, you can create neural activity where it doesn't exist, such as artificial vision for blind people, or light-driven pacemakers for people who need more regular electric activity in their hearts. So how does it work? Basically, we use various biological means to introduce these rhodopsins into neurons, and then they can change the electric potential of neurons by passing ions actively or passively and suppressing or activating them. So optogenetics is a very exciting field. It garnered lots of prizes in the last 2 years, very important international prizes. So optogenetics keeps developing. So what I showed you on the previous slide was first generation optogenetics, which relies on changes in the membrane potential. Now here is the second generation, where you rely on changes in intracellular signaling molecules, such as cyclic GMP or cyclic AMP. So you can use enzyme rhodopsins to create cyclic GMP, as shown here, or to destroy cyclic GMP, as shown here. So these are all fairly recently discovered and characterized molecules. Great. So this will finish my introduction into the microbial rhodopsins. I hope everyone agrees these are very exciting molecules. Now I will try to tell you what we are doing in our lab. And basically, the main goal of our research is to understand how is it possible to take this simple 7 helical template with retinal and to change it in a way that it creates so many different functions. I showed you so many different examples, right? Many different functions, but they're all based on very similar proteins. So it doesn't take much. We want to understand how these proteins work and how you can interconvert them, create different functions. We also want to find new functions and to study them with the help of physics. We want to understand all the detailed molecular mechanisms. And in the past decades, we characterized many new rhodopsins, some ion pumping, some sensory, from various sources, and we combine molecular biology, biochemistry, and physics. So I will show you our interdisciplinary pipeline. So our group is very interdisciplinary. We have biologists, chemists, and physicists, and it is reflected in this pipeline. Basically, we are trying to do everything in-house. Of course, we have a lot of collaborators, but we combine a lot of different disciplines, even though we are in physics department, but we are doing some biology as well. So here is our pipeline. This is how we work, and I will describe some methods and give you examples, and then I will finish with a more extended example of a project. So we start with finding some new genes. This is so-called bioinformatics. So we are doing searches, we are trying to find something new and unusual, and then we try to characterize it. To characterize it, you have to make it. So we make it by getting the DNA, and then we employ these microbes, such as E. coli, very common bacterium, or yeast, so-called Pichia. So we force them to make our proteins. Then we have to isolate them. We break them, we break their membranes, and then we put our proteins back into the lipids. This is important because these are membrane proteins. They like to be in lipids. So after we purify them, we put them back into lipids. And then we use the array of physical methods here on the left. These are done in our labs. And here at the bottom, there are all kinds of collaborations we have. We have collaborations on other techniques, which I'm not going to talk about much, but we do have lots of help from other groups to complement what we do here. So I will start with, well, we will be moving down the energy spectrum of electromagnetic spectrum. So we will start with the most energetic photons, which are UV and visible. And you are all probably familiar with spectrophotometry. It's very simple. I will give you one example on visual rhodopsin. So you can take spectra in the UV vis of rhodopsins, and if you are lucky, you can catch activated states without doing time-resolved spectroscopy, just doing static spectroscopy if these states are stable. So here is an example. Here is your visual rhodopsins, which we have in our eye. When you shine light on it, it changes from a red pigment to yellow pigment, which absorbs in near UV. And this is actually the active state, which signals to your brain. This is simple, of course, still can be very useful for studies of your proteins, but we are doing something a little more sophisticated, which is so-called time-resolved spectroscopy in the visible. Imagine that this transition is very fast, and we cannot catch it by doing static spectroscopy. Then we need to do some time-resolved spectroscopy. Time-resolved spectroscopy is a pretty old method. It is called flash photolysis. So this is a typical setup for flash photolysis in our lab. So basically you have a light source, which is a measuring light source, the monochromator. Here you have your rhodopsin sample. Here you have another monochromator and detector. And here you have a laser. And so the laser excites your rhodopsin, and then you detect the changes at a single wavelength. And then you can change the wavelengths and measure another. And By strategically selecting the wavelengths, you can study a lot of biological processes. So you should, of course, know where to measure, but it is surprisingly rich in information. Here is an example. This is bacterial rhodopsin, this prototypical protein I introduced, the proton pump. And this is a typical flash photolysis curve. So here is a logarithmic time scale. At time 0, we have a flash of a laser flash, green laser. And here we are looking at the difference. This is difference spectroscopy, right? So if signal is positive, something is forming. If signal is negative, something disappears. And so I give you just a few examples of how following these kinetics at different wavelengths can give you lots of information about the protein. So for example, if you measure at 410 nanometers, you can see deprotonation and reprotonation of the active center of bacterial rhodopsin, of the retinol. Here you can see isomerization, the change in the shape of retinol. You can also add a pH-sensitive dye, and you can actually see in real time how your protein spits out protons and absorbs them back. So, fairly powerful method, even though very simple technically. Next, many of our proteins pump ions. They pump chloride or protons or sodium or other ions, and we use special assays to test this, whether they're pumps and what kind of ions they pump under illumination. So I will give you an example. Here is how we do it. So we form so-called liposomes, which are enclosed vesicles, lipid vesicles, and we insert our proteins into these vesicles, and then when we shine light, they pump protons in or out, depending on what kind of protein it is, and then we can detect it. We can detect it by measuring changes in the pH of the solution. And then we can check if the transport is active by adding a proton uncoupler. So this example is from our discovery of a first proton pumping protein in fungi. Okay, let's move down the spectrum. Weaker, less energetic photons, the infrared spectroscopy. So the infrared spectroscopy, of course, very powerful. Checking the vibrational spectra gives you lots of information about different groups. So let's discuss it a little bit. So many of you may be familiar with static infrared spectroscopy, which is shown on top. So static infrared spectroscopy of proteins is used widely by biochemists to check structure of the proteins, because these so-called very intense amide bands are sensitive to the structure. They will tell you if your protein is helical or not, and you can follow your protein conformation in overall terms. You cannot get very detailed site-specific information from the static spectra. But what we do, we use so-called reaction-induced difference spectroscopy. What does it mean? We take a spectrum before illumination and after illumination, and then we take a difference. And when we take a difference, we can see only those groups which are active, right? So here is an example from another protein, not from our group. I just like this cartoon. It's very nice. So it shows you that when you take absolute spectra of two states, they are very similar. There is not much you can do. The difference is very small. But if you magnify the difference, you can see signals which can correspond to single atoms, literally single groups being protonated or changing their vibrational properties, right? So you can see pairs of atoms with this spectroscopy. So some people cannot believe it. They say, okay, this is really large absorption around 1. Here you're looking at absorption of on the order of 1 milliod. But it is real. You just need to average a lot. So we usually average a lot of different spectra to get reliable information. So this can be done, different spectroscopy can be done statically, or it can be done in a time-resolved mode. And then you have a 3D picture, you have spectra, and you have kinetics. And I will give you some examples on this. Here is how the different spectra can be useful to follow the protonation. So I told you many of our proteins are proton pumps, so we need to look at the protonation changes, and carboxylic acids protonate, or they change their hydrogen bonding. And all of this produces signals in the infrared, right? So the different spectra, if something, for example, deprotonates, you will get a negative signal. Here is the explanation. And if something shifts, you will get negative and positive signal. So I will give you another example on rhodopsin from our old work. So here is a different spectrum, a snapshot at, let's say, several milliseconds after the light excitation of bacterial rhodopsin. And it just shows you how rich the spectrum is. You can see overall conformational changes by looking at the amide bands. You can see protonation and deprotonation of carboxylic acid, so you can follow the traveling of protons inside the protein. You can see how retinal changes. Here, these are bands responsible for reporting on the retinal isomerization, and here are bands reporting on planarity, whether your retinal is twisted or flat. So lots of information, and we can take a lot of these snapshots at different times. So we use this extensively on our own and in collaboration. This is a very powerful technique. You can assign specific bands very often to specific groups by using mutagenesis in combination with infrared spectroscopy. Okay, the next type is Raman spectroscopy, which is also vibrational spectroscopy. It is complementary. Probably all of you heard about Raman. I will remind you the idea. So basically, when your protein scatters light, if it scatters the same energy, it is well-known Rayleigh scattering, elastic scattering, but some molecules rarely, very rarely, will pick up or lose vibrational quantum. And this will give you the vibrational spectrum, very similar to the classical infrared absorption spectroscopy. But the selection rules are different. So you may see different shapes, and you may detect different groups when you compare Raman and infrared, even though they are both vibrational spectroscopies. More interestingly and more importantly for our proteins, there is also such an effect as resonance Raman. Resonance Raman is when you have electronic excitation, and when it relaxes, the probability of Raman process increases dramatically. What does it mean? It actually means, imagine I have a large protein and I have retinal chromophore which absorbs light. And the resonance enhancement actually enhances signal from the retinal, but I don't see the rest of the protein. This is good and bad. With infrared, I see the whole protein. With Raman, I only see the retinal. So if I want to study retinal, Raman is my method. If I want to study carboxylic acids, then no. Then I should do infrared. Here is an example from our lab. We have FT Raman machine, and it shows spectra of two different states of a rhodopsin. And it shows you that different conformations of rhodopsins, this is all trans, you see it's all flat, and this one is isomerized, and they will have very different fingerprint spectra. These fingerprint spectra are carbon-carbon stretching vibrations, and I will know what happens with my protein when I shine light on it. I will know exactly what happens to retinol. So this is very powerful. Okay, the last method, and I'm going to talk about it in greater detail, because this is what we are doing lately. We are spending more and more time on this compared to other methods, because this is extremely powerful method which can give you a lot of information at the atomic level. And I should say this is all done in collaboration with my good friend and colleague, Professor Ladizhansky, in the same department, and most students we co-supervise and work on joint projects. So we have two labs which form one powerful supergroup, which combines all these biological and spectroscopic techniques with solid-state NMR. So I will give you some examples on our NMR work. So first, an overview. This is our 800 megahertz magnet. We have very good NMR center at Guelph, and we are doing it in solids. So, right, there are two main types of NMR, as you probably know, solution NMR and solid-state NMR. And because our proteins are very large, and they also reside in lipid membrane, so the samples can be classified as solids, not as solution. So if you know a lot about solution NMR, some things will not apply to solid state. I will explain very soon. So basically, what can you get from solid state NMR? If you're lucky, you can get information about every atom. You can get information about environment of every atom. You can also get information about how atoms are located relative to each other. So you can measure distances between specific atoms. So you can get secondary structure of your protein. You can get full structure of your protein if you're lucky. You can study how proteins interact with each other. You can say things about the environment of each atom, and you can also measure their dynamics. You can measure how fast they move. So this is extremely powerful technique. It can be done in lipids under native conditions. So it is very, very powerful. So there is one caveat, which I want to mention from the outset. This is where we need biology, because we are doing carbon and nitrogen NMR. And the isotopes which we use should have spin 1/2. And the natural abundance proteins do not have this. So to be able to do NMR, we need to isotopically label our samples. And we do it biosynthetically. This is where biology is so important, because we introduce carbon-13 and nitrogen-15 isotopes into our samples. Before I give you some examples, I want to remind you a few basics so that you have better appreciation of the example. So I will remind you, two main things which we should keep in mind are chemical shifts. So chemical shifts basically are signatures of each atom. So each atom has, each nucleus has its own resonance frequency, which we characterize by the chemical shift. Right? So as you remember, the principle of NMR is that we put molecules in very strong magnetic field, B naught, and then spins, carbon-13, nitrogen-15, and protons orient against or along the magnetic field. And the energy difference can be probed by supplying radio frequency pulses, which come into resonance with this energy gap. So the chemical shifts occur because each atom, even if it's carbon-13, is different. because they have different electronic shielding, which reports on the environment, which reports on the chemical structure. So this is why we can distinguish every single atom, because they all have slightly different electronic structure. And this is great. So we can have signature for each atom, and we can also get information on the environment and also on the structure. We can get dihedral angles of bonds between atoms, which is very important for proteins. The next important physical phenomenon is dipolar coupling. So spins, little magnets, talk to each other through space. This is called dipolar coupling, and which is dependent on their mutual orientation and on the distance. So when you do solution in MR, dipolar couplings are not that important because they average out, because your molecules tumble in solution. But in solids, they do not. They do not tumble. So you would think your spectra will be very broad and messy because there are all kinds of orientations present in your solid samples, powder samples, and you will not be able to see anything. This is true. So if you look at static spectra of powders, they are very broad. What we do, though, we use so-called magic angle spinning solid-state NMR. So we pack our samples into these little rotors, which are spun at very high speed, let's say 12 kilohertz, 14 kilohertz, or higher, depending on the kind of NMR you're doing. And then you average out these dipolar couplings and you have very nice spectra. You don't want to eliminate all the dipolar couplings, of course, because you use them to measure distances. So we reintroduce them selectively after averaging out most of them. Okay, so this explains the two main things we use from NMR: chemical shifts and dipolar coupling. So the environment, structure, and distances between the atoms. Great. Now I will give you some examples. So my example will be on our favorite protein, which we spent the most time on. This is so-called anabenesensory rhodopsin, or ASR. It's a very interesting protein from a cyanobacterium, which was discovered back in 2003. So rhodopsin interacts with the transducer. When you shine light on it, transducer leaves and interacts with DNA. And so it regulates activity, photobiology of this cyanobacterium. Okay, so we wanted to study this protein, and we started with all kinds of traditional spectroscopic techniques, but then we switched to NMR. We realized we can get a lot of information, and I will give you some examples. So here is how it looks. Picture better than 1,000 words, as you know. So these are bacteria in which we grow this rhodopsin. These are E. coli. Then they look beautiful, right? They're colored because they have lots of rhodopsin. Then we break the membrane, use detergent to break them, and this is our protein in detergent. I told you we want to put it back into lipids, so we mix it with lipids, and then we withdraw the detergent by so-called biobeads, which are porous beads, and then we have these proteolipid complexes, which we put into these rotors for magic angle spinning. These are, of course, N15 and C13 labeled. And before we do NMR, we can do infrared spectroscopy to double check that our protein is healthy, it has enough lipid, because in infrared we have signatures for all of this. Great. Then we started doing NMR, and NMR turned out to be amazing. The very important thing in these projects is to have very good spectral resolution. So you see every single dot here is a pair of atoms, right? These are so-called two-dimensional spectroscopic plots, which show you correlation between atoms. So magnetization is transferred from an atom to its neighbor. So every atom which is within 5 angstrom from each other will be on this spectrum. So you should have very good quality of sample to be able to see this, because the order of your sample is very important. But we were lucky, so we have very good resolution. And if you have very good resolution, you can do the next very important step, which is called assignment. So the assignment is when every single one of these dots, or almost every single one, is correlated with a pair of atoms. So we were able to get 206 assignments. So we have 229 amino acids in this protein. And so we have a spectroscopic handle for most of them, which is amazing. It allows us to study proteins, this protein with NMR, at the atomic level, because we do have a spectral signature for each. First outcome is that we could see the structure, right? We could see the structure, but looking at the chemical shifts indicative of the structures. I told you can get torsional dihedral angles, and that tells you about the shape. So we could see all the helices here shown with positive indices, and we could see some other structures in the connectors of these helices. This is not super exciting, but at least it told us that we are doing something right. We reproduced the 7 helical structure, and then we measured the distances, and we tried to calculate the structure. So we combined this dihedral angle, so-called torsional constraints, and distance restraints, the distances which are on the order of 5 angstroms or less, and there is a computational procedure to calculate the structure. So we calculated the structure, but we were not very confident this is the right structure. Why is that? This is because these proteins like to talk to each other. So we suspected that some of the distances we measured may be between atoms in different proteins, not within a single protein. You see, it's a problem for the computational procedure. So we needed to know what kind of complexes these proteins form. And this is what we did. We now use paramagnetic labels. So paramagnetic labels, such as an MTSL, are based on nitroxides, and they're used in electron paramagnetic resonance spectroscopy. But we also can use them in the NMR experiments. Why? We can attach them specifically. We have biochemical means to attach them in only one place. We choose by introducing special amino acid cysteine, which reacts with this label. What happens is that NMR spectra will broaden for every atom within a radius of 15 angstrom. So here is an example. So these are intensities of NMR signals. Here, this is where I put my label. So, not surprisingly, the amplitude went down. But then we see that the amplitude went down very far from it, and here as well. So what's going on? Well, hard to understand without looking at the structure. So let's look at the structure model. So we look at the structure model. So these are 7 helices. This is the label, the paramagnetic label, and in green you can see the residues which are attenuated. It doesn't make any sense, right? Here is a label, here is attenuation around it, that's just fine, but then there is attenuation very far, but no attenuation in between. So it's impossible that the attenuation travels so far. It doesn't make sense, but it makes sense if we recall what we wanted to see. We wanted to see that whether this protein forms complexes. And so that's what we did. If you now keep in mind that this is a trimer, similar to some other rhodopsins, everything starts making sense, because now you can see that the labels actually talk to atoms, quote unquote, atoms on the neighboring monomer, and then everything makes sense. So this attenuation here comes from the neighboring monomer, not from this label, but the label on the neighbor, like here. So this was a very exciting moment. We realized, oh, we now can actually explain everything if we assume that it is a trimer. So complex of 3 proteins. So we assumed that, and then we verified it, of course, by other spectroscopic techniques. So here is a circular dichroism. I didn't talk about it. It uses circularly polarized light, and basically it is sensitive to interaction of the chromophores. So whenever you have trimers, you usually see this bilobe shape, and that's what we observed, both in detergents and lipids. Then we also used biochemical techniques, classical biochemical techniques, which can verify the oligomeric state. Okay, so after that, we could sort out the distances which we measured as intramolecular and intermolecular, between the molecules and inside the molecules. And we could recalculate the structure. And so we recalculated the structure, and this is what we got. So this is the structure which we calculated from NMR, which was basically the first structure to get by solid state NMR for rhodopsin and lipids, and maybe the second structure for a large membrane protein. And we could also see how it interacts with its neighbors, right? So we could see the structure of a full trimer, not just a single protein, but how it forms the contacts. Later on, we decided to improve our model, and we collaborated with people who are doing electron paramagnetic resonance. We used the same labels which were used for paramagnetic relaxation enhancement. Now, our friends in North Carolina did Deer spectroscopy. This is where you're measuring dipolar coupling of electron spins on the labels you introduce. And this modulation is very sensitive to the number of spins. So they could verify we have trimers, as you can see here, and they could also improve the model. Finally, these rhodopsins, some of them form 2D crystals, as you know. And we could verify that our rhodopsin also forms a 2D crystal by doing small angle X-ray scattering. So another collaborator did small angle X-ray scattering for us, and he saw very nice Bragg peaks, so we could model the lattice. So it's not just a trimer, it's a lattice of trimers, very similar to bacterial rhodopsin. Okay, the last newer project I wanted to describe is related to the use of deuterium to look at folding and conformational changes of our rhodopsin. So the principle is very simple. You can exchange exchangeable protons for deuterons, and the exchange will only occur in the exposed parts, but not in the buried parts. And this way you can look at folding, stability, and conformational changes. So for NMR, it's very simple because in the simplest version, you just start your measurement with by transferring magnetization from a proton, but if it's replaced by deuteron, you lose the signal. There is an alternative where actually, which is the opposite, where protons actually destroy your signals. So these are two complementary techniques. Either way, you can detect what is exchangeable and what's not. And in the past, we did these experiments with ASR, looking at the conformational changes. We did hydrogen deuterium exchange in the dark and upon illumination. And so what's exchangeable in the dark is shown in blue. Not surprisingly, these parts which stick out of the membrane are exchangeable. But then we observe that when you shine light, solvent starts to penetrate here into the cleft. They said, oh, great, we can now picture the conformational change, which is induced by light. That was very exciting. We thought we could see the opening of the protein shown by red. Yeah, so now we turned to something else. We want to see the unfolding, and which will help us to understand the folding of this protein. So how we do it? We combine thermal stability assays with solid state NMR. So basically we heat up our protein in deuterium, in heavy water, it penetrates and it exchanges with protons, which we can detect as I just described. And then we cool down and measure. And we heat up at higher temperature, cool down and measure. Again, it should be reversible. We shouldn't overdo it. If we heat too much, it will be just destroyed. But we are keeping ourselves in this reversible regime. And what we did it at different temperatures, and we got some very interesting insights, which I will summarize on the next slide, which were published a couple of years ago already. So here is the picture of solvent penetration, if you want, at different temperatures. Here you can see at room temperature, solvent is mainly outside. Then you heat it up to 76 degrees. You see much bigger penetration. And then at 80 degrees, it's even bigger. But we could identify the stability core. This core around the retinal chromophore does not exchange, right? We could also see that these places where proteins interact with each other are also very well protected. They do not exchange even at very high temperature, 80 degrees, right? So it's shown here. And we could also see that one of the helices, helix number 7, exchanges a lot. So we thought that it is the weak link, so to say. This is a weak spot, which folds the last, unfolds the first. So lots of mechanistic insights. And I repeat, we could only do this because of all the preceding work, early work, which I showed to you, because we have spectroscopic signature for virtually every residue in this protein. Okay, with this, I think I'm running out of time and I will finish. I hope I gave you some overview of what we are doing. Very interdisciplinary, but I believe it is exciting. We are doing lots and lots more, working on all kinds of new rhodopsins, but this may be for the next time. So I want to thank you for your attention. I also want to thank all the members, past members and current members of our groups of Brown and Ladejansky. I want once again to thank Vlad, who is our NMR guru. And I also want to thank all our collaborators, and there are many. I couldn't talk about them, unfortunately, but there are many in different countries. And I want to thank our funders, of course, as usual, NSERC, which is a Canadian agency for funding this basic research, and as well CFI, which gives us very good money for infrastructure so that we are able to buy all these wonderful spectrometers, and of course the University of Guelph, which supports us. And thanks very much again for your attention, and if there are any questions, I will be happy to answer them.
Thank you, sir, for your wonderful presentation. So we have got few questions in the inbox, not in a comment section. So the first question: what is the thermal unfolding of ASR?
Sorry, could you please repeat?
Okay, I can add the question in the chat box. So that you can. What is the thermal unfolding of ASR?
I'm going. Oh, what is thermal unfolding? So basically, as you all know, proteins have thermal stability. So why, for example, fever is so dangerous? Because some of your proteins are not designed to work above 40 degrees, right? So because certain interactions break and proteins change their shape. So ASR has very high point of thermal unfolding, about 80 degrees, compared to human proteins, which don't like to be above 40, right? So yeah, basically it's breaking of the structure, change of the shape, helices come apart, but it's a very actually not trivial question, because to understand what is the unfolding of membrane proteins is difficult. They do not unfold completely. It's just some partial unfolding.
Yes. There is another. What is the lipid embedded A, sir?
Yeah, so these are membrane proteins. We study proteins which are membrane, right? They reside in our cellular membranes, which are made of lipids. So we should put them back into lipids, and that is what is called lipid embedded, because they are surrounded by lipids. So we try to recreate their native environment.
So this may be the last one.
Yeah, what is retinal chromophore? So the retinal chromophore is this light-sensitive cofactor which is attached to our proteins and which gives them the light, ability to see the light. It gives them the color. This is why it's called chromophore from Greek, giving the color. But it is the most important light-sensitive element of our proteins, important for vision. We have retinal chromophores in our eyes, and bacterial proteins have retinal chromophores.
Okay, sir. So thanks again for giving us this time, and hopefully we can arrange another webinar with you after a few times, and if you have a time. So thanks again, and we'd like to say thanks again on the behalf of the Department of Physics, Pabna University of Science and Technology. So have a nice day, sir. Bye for
Thank
today.
you. Thank you very much, everybody.
Okay.